So, welcome everybody. This is the penultimate kind of event. We're calling it an open mic for data for the BIOFAIR Data Network project. So, my name is Matthew Sheik, and I've been working with the Beacon Steering Committee for the last 2 years on this project. And so, I'm just going to give some background of the project, some housekeeping, and then kind of give you this concept of what we're doing here today. Okay, so the first is just some housekeeping. So we have 10 lightning talks, 5 minutes each, and we're going to have a discussion section after all of the talks. And so we ask that you use the chat to either record your questions and we'll um, answer them after, or if you just hold your questions, then you can use the raise hand function and the discussion facilitators can um, call on you. Other things, this session is being recorded and will be posted on the Beacon website after the event. So if you have to step away for 10 minutes or whatever and you miss something, you can come back to that. And all of our speakers have given us permission to record their section of the presentation and share their slides afterwards. So those will be available too. If there's any kind of technical difficulty, we have some IT support in the chat. So go ahead and shoot a message there and we can get those questions figured out for you. Okay? So, just some background on the project. We've been doing this since spring of 2024, and the BioFair Data Network project summatively was a project aimed at so the last decade and a half we've spent digitizing and accumulating biodiversity data, and the next steps basically we're trying to kind of reach out to the communities. Specifically, we had 6 domains, all the way from federal agencies to One Health to biodiversity data to environmental and ecological data, and basically gather these folks from domains that are not specifically the natural history collections communities to see how we can move forward with all of this accumulated data to create this integrated and open fair data network that connects all of these things. And to do that, in last year in the summer and the fall, we held 6 virtual listening sessions. These were about 2 hours each, and each of the 6 were one of the domains. And then after those listening sessions, we held a inclusive all-day virtual workshop to basically develop recommendations and figure out potential next steps towards building this grand idea. Um, and then this past summer, we spent kind of synthesizing all of that into a final report. We have a roadmap, and we did um, basically some webinars, and then we also have a report. And so now this is kind of like I said, the penultimate. So this is not your typical webinar. I'm going to try and give it a little spiel, but basically we thought, if we want to instigate cross-domain conversations, a typical webinar setting is not going to work for us because we don't want to just talk at you. We want to have these discussions. And so we kind of formulated this idea that this is more of like some kind of open mic situation where we can have folks step up to the mic, and then we can also have folks in the audience who are there with their ideas and their discussions. So after the talks, I highly, highly encourage you unmute and talk about some work that you might find fits into this broader roadmap that I'm about to show you. Um, and we hope that this is a space where you can get ideas about projects that have to deal with integrating various data, and also potentially find collaborators, or just get, like I said, just get some good ideas. Okay. So This is kind of like this figure is the summative of the many, many, many hours of community discussions, internal deliberations, and things like that. And so what I want to point out first is that I have two QR codes on the screen. The left one is a podcast that we recorded, basically discussing the project. And so if you want a quick 20-minute, 25-minute listen about what this project entails and our findings, and what the community has kind of come to a consensus about, then I would recommend listening to that podcast. And then we also I have a QR code on the screen so that it links directly to the report that you can read as well. So basically, what we have kind of extracted from the many domains of that have participated in the BIOFAIR Data Network project is that there are really two general fields of where we can move forward. There's a technological aspect and there's a social aspect. So for the technological aspect, it's really about this idea of our siloed systems needing to come together. So we have to do things like stock take and do some gap analyses. For instance, one example that came up during discussions was the use of like defined vocabularies, and are there defined vocabularies across domains that can actually be kind of integrated into each other so that there are definitions from one field that help define data that haven't been defined in another field and vice versa and things like that. But then also there's that actual like capacity building where we have to create these tools that help link these silos. And then the other aspect is the social dynamic of this, where there has to be this cultural shift, right? Of this collaborative mindset. And so we have to do things like educate and train folks on how to even manage their assets, their data. We also need to come together and establish like sorry. We also need to establish best practices and standards. So we have tons of best practices and standards, and really what it's about is integrating them all together to kind of create, instead of having these disparate best practices, we have kind of this community consensus that we can build through community input and things like that. And I'm saying community a lot because that's also an aspect of this, where we need to come together and do community building, not only within our domains, but across domains. So, for instance, an example that stuck out to me during this project was this idea that you can be really technologically savvy, but there are folks out there who are trained and learn about pedagogy and how to teach. And so partnering with those kinds of partnerships can really bolster this idea of creating and developing like like this cross-disciplinary training, and things like that. And so these lightning talks that we have today, basically, these speakers find themselves in one of these chunks, or multiple of them, and they are here to basically highlight work that is kind of already biting at this pie, basically. So the Beacon team really has come to understand that this is a really, really big endeavor, this BioFair data network. And so really the first steps are just starting to take bits and pieces that we can chew and kind of create a collective. If everybody takes a bite, then collectively the pie will get eaten eventually, right? So with that, I'm going to turn it over to Brita to introduce our speakers.
Thanks, Matthew. I'm going to make very quick introductions for each speaker, and I apologize in advance if I mispronounce anyone's name. As Matthew said, we have a shared deck of slides, so you'll just need to unmute yourself and ask Matthew to advance to the next slide. I will also unmute myself when you hit 5 minutes and ask you to wrap it up just to keep us on track with time. And lastly, for everyone on the call, if you can hold your questions until all the presentations are done, then that would be ideal. We can incorporate them into the discussion. So first we have Donat Augusti from Plausi, who will talk about a taxonomic name is the key to the knowledge about a specimen.
Okay, thank you for inviting me, sitting here in Lucca on the other side of the Atlantic. I'm president of the Swiss NGO Plausi, whose mission is to liberate data imprisoned in scientific publications. So today I will talk about what's in a taxonomic name and why it's important to link to it properly. So what is a taxonomic name? It is and has been an identifier for a taxon. It refers to one or multiple linked taxonomic concepts, like the cascade of synonyms, for example. It enables comparing taxa-related data over space and time. It allows reproducibility, so an original description includes the evidence why and on what basis a species has been described, and it has a link to holotype. It is the provenance in the case of an identification, so you can go back and say, why did he or she decide to use this name? And more importantly, it's part of the biodiversity knowledge graph. So it's in science we cite, and the taxonomic name and the treatment is full of references. So it's important in the reproducible science. It can be very operational if all the links are really made. So you don't need to go and find any old literature. It's here because it's next to the mouse click. And if you want to know more, follow the Disney's roadmap, a decadal roadmap to liberate data and imprisoned in scientific publications. Next slide. So if you visualize this, a taxonomic name has always a taxonomic treatment, a well-defined section in a scientific article. It is based on observations, which are represented as material citations, and those are linked to the original specimen, like a holotype or other material. They're linked to institutions hosting the specimens, to a collector, and so on. Next slide. So how do you make them fair? So in the middle, you see one, when we get a publication, we have the treatment bank factory that's part of the Plausi ecosystem, which opens up, as you see on the left, a PDF or any article or a book. It liberates the data therein, the references, the images, tables, treatments, material citations, and makes them fair. That's number 3, together with Zenodo's biodiversity literature repository, where we created fair digital objects for treatments, figures, and which then can be cited and can be reused. We make this data all accessible. We actively collaborate with, for example, the Global Biodiversity Information Facility, which uses the data, material citations, the treatments, and the figures, and currently the publications are the largest data set provider in GBIF, with almost 50% of data sets. We are cross-domain, so we move from green to blue. That's life sciences, where we build together with the European PMC, the biodiversity PMC, which allows cross-domain searches among life sciences and the biodiversity. Next. Just to show this is not just a fluke. So we started more than 10 years ago building treatment bank. And in the meantime, we liberated data from 128,000 publications, which resulted in 1.2 million taxonomic treatments, 780,000 figures, 100,000 tables, 2 million material citations, and close to 5 million bibliographic references. And they are inaccessible, either in treatment bank, which is a data lake, or more and less in the biodiversity literature repository, GBIF, the biodiversity PMC, or in checklist bank. And you see the number drops, and that's because of quality control. So there is a step from automated liberation to make it really a curated data set. So go there, look at this, make use of it. They're all there, citable. Thank you.
Thank you very much. Next up, we have Jason Best from the Botanical Research Institute of Texas to talk about exploring Wikibase as a platform for the Sherwin-Carlquist Extended Specimen Network.
Hey, great. Thank you. And good morning, good evening, afternoon, wherever you are. Um, as she mentioned in the title, I'm here presenting some collaborative work we're doing to create an extended specimen network, an ESN, for Dr. Sherwin Carlquist. He was an American botanist known for his work on island biogeography, uh, on wood anatomy, and also for his prolific use of photography to capture field images. The project is a collaboration between the Botanical Research Institute of Texas, BRIT, and California Botanic Garden to unite the various research products and assets that he created throughout his career. The archival items are at BRIT, digitized and deposited at the portal to Texas history. His specimens are at CalBG. They're digitized and deposited at cch2.org, a Symbiota portal for California. The goal of this project is to connect these specimens, field images, notebooks, and other items across these different platforms and create an extended specimen network. And people familiar with this have probably seen this diagram many times, but it represents the physical specimen at the hub or the core of this extended network with various levels of extensions, with the first being the digital record and media of the physical specimen and extending outward to field notes, field images, etc. Next slide, please. So we were able to make this connection making links between the two portals. In Symbiota, for each record that had a match, we were able to create a link within the interface that links to images on the portal to Texas history. So for example, this has two images created in the field. On the portal to Texas history, we also have metadata, including links that are reciprocal and link back to the physical specimen record. These links are navigable by humans. It allows a person to explore it more, to enhance the access and discoverability of these records. But these links are meant for humans. Not really. They can be navigated by a machine, but they're not really interpretable and actionable at the machine level. So next slide, please. We wanted to explore different ways we could go beyond the human accessible ESN and extend it into the linked open data universe. We started looking at Wikidata and Wikipedia as a way to approach this. On the left, you're probably familiar with what a Wikipedia record looks like. It has a natural language description of Sherwin Carlquist. It's written for humans. It's organized, but not really structured. On the right, you see the same a record for Sherwin Carlquist, but it's much more structured. It has defined properties, defined values and fields, and it's more readable and accessible by a machine and it's more structured like a database rather than just natural language. These bits of information can be linked internally. As an example, the ones you see in blue and underlined are linked to records, for example, to BRIT, where his archives are, to CalBG, where his collections are. Those are all information that are accessible within Wikidata. But you can also link out to other resources like the BHL Creator ID links to the BHL record for Carlquist and Bionomia ID, etc. Next slide, please. So we explored this a bit. We did some work in Wikidata to make some additional linkages, but we also created our own private Sherwin Carlquist Wikibase instance. It's a standalone free instance that we are exploring these concepts with and have started populating this we uploaded multiple records of the physical specimens into this Wikibase instance. We uploaded BHL article records into it, and we started making these connections within our private standalone Wikibase ESN with the hopes of seeing how it would work to link from the public Wikibase into our ESN and out from our ESN back into the public Wikibase. Also to linking out to articles in BHL, linking out to the ESN we had already created on our separate platforms, linking out to the images in the Portal of Texas History, field images and notebook images, linking out to the physical specimen records at CCH2. And more importantly, making a platform that is accessible and can be integrated with broader linked open data resources, and making these resources more accessible for machine reading to be able to query across multiple platforms and making this information more broadly accessible for humans and machines.
Jason, 5 minutes.
That's it. Thank you very much.
Thank you. Next up, we have Rachel Blake from the Intertidal Agency to talk about building the culture of data stewardship and ocean data case study.
Thank you. Hi, everyone. I'm Rachel Blake. I'm Director of Data Science at Intertidal, and I'm happy to be here to share with you a bit about our work on data stewardship. So we are a nonprofit working to unlock ocean data for global good. And we really describe what we do as building the people layer of data sharing and data governance. This can also be called the human infrastructure part of data ecosystems. And I think Matthew earlier on this call referred to it as like the social cultural aspects. So we're not building new tech widgets or apps or designing access controls, but we really believe that that sort of technical infrastructure should be built in support of the people, the policies, the processes, the workflows, and the standards that enable and really make possible fair data integration and data reuse. Next slide. We see data stewardship as really as a key to unlocking a whole new bunch of data discoveries for ocean ecosystems. New innovations and new long-term sustainability and conservation efforts as well can be unlocked through data stewardship. And really, without good data stewardship, the future us, so future data professionals and research professionals will struggle to understand existing data in context, to preserve data for future analysis, and to integrate data to answer new questions. And so this is across domains and sectors as well. Because we see data stewardship as such a critical thing, we're working to build this culture of data stewardship across ocean sectors. So we're working with other nonprofits, federal and state government, and academic research as well. Next slide. So at Intertidal, we really do believe that everyone, no matter your role working with data, can practice good data stewardship. So maybe you're not the one analyzing data, but maybe you're setting up the policies and the environment where data can be integrated and reused. So these sorts of actions that make up good data stewardship can include planning for data management and data publication, documenting data by creating robust metadata, standardizing data for more streamlined integration and through workflows, and increasingly through automated machine actionable workflows. Licensing data to clearly communicate permissions for reuse, and supporting discovery of data through multiple platforms and ways of searching for data for new projects. So we really try to meet people where they are on their data stewardship journey and help build the habits and the skills that support fair data. Next slide. So I'm here today because we really want to collaborate with other folks. We want to build this community through training for data stewardship. So we'd really like to hear from others that are teaching these sorts of skills, how you're building community around similar topics in your work. We have collaborations with Creative Commons to teach about data licensing, with OpenScapes to teach open and reproducible workflows and scientific practices, and other organizations to teach about metadata standards and data publication. We've even done workshops on specific metadata standards like Darwin Core and really sort of gotten into the nitty gritty of some of these topics. So while we're focused on the ocean domain and ocean data, we make our work generalizable and reusable as much as we possibly can. So if you're interested in sharing ideas about data stewardship lessons or methods or workflows and how to build this community around these sorts of things, we'd really love to hear from you. We'd love to collaborate with you. So my contact information is on this slide here, and you can find us online, and look forward to chatting. Thank you.
Thank you very much, Rachel. Next up, we have Israel Borakini from Montana State University at Bozeman. The title of his talk is Eco-Evolutionary Analyses of Global South Biodiversity Need Better Data.
Hi, everyone. Thanks for inviting me to this talk. Today, I will be talking about the Global South biodiversity and how it can be harnessed to do more analysis to understand how the drivers of biodiversity in these really mega diverse continents. So we all know about the longitudinal diversity gradient. As you move towards the equator, biodiversity for most taxonomic groups are really higher there, which is mostly driven by both ecological and evolutionary drivers. Next. But at the same time, there have been so many papers and publications that have come out to highlight the fact that there's so much data gaps in for these regions, as you see on those slides, with areas close to the equator, having fewer knowledge, data availability for doing robust analysis to advance science in this particular area, despite the fact that we keep saying that these regions have the highest biodiversity. It's also the region where we know the least. Next. And so we might begin to ask yourself the question, where exactly is the biodiversity data for the Global South? Where is it located? So first, next. It's found mostly in the Global North museums and Iberia, which hold most of the collections from Asia, tropical Asia, Africa, and South America, which is where most of these collections are. And but unfortunately, many of these collections were made centuries ago, and they have no georeferences, so which limits their uses. Tons and tons of data in Kew Gardens, in Paris, in Brussels, that have no coordinates, and so they are just sitting there with very few limited uses for them. Next. Some have been digitized, but many have not been digitized, and that will make me to give a shout out to a project that is currently ongoing in the US, next, which highlights the digitization of millions of specimens of African plants that have been managed in various herbaria in the United States, and this project is being funded by the NSF. Next. Secondly, there are many, of course, biodiversity data is found in nature. It's still in nature, and they are begging to be discovered, to be explored, and to be surveyed. And most of this is being limited due to poor funding. Next, and finally, there are many herbarium records as well, especially for plants that are sitting in shelves that are yet to be mounted, processed, and digitized in herbaria all over Global South. I just came back from a conference in Africa where this was a common theme, with millions of specimens being collected, but yet to be mounted, digitized, or processed because they have no resources to get the basic materials they need to process these specimens. And so this is where biodiversity data is sitting. This is where they are, and that's why we keep complaining that there's so much data gaps in the Global South. This is where those data are seated. Next, next slide. And so, but when we have these biodata, what do we do with them? My work involves identifying, using spatial phylogenetic tools to identify centers of phylogenetic endemism and diversity, next, which can be used for conservation prioritization and gap analysis. As you see on the slide there, those areas in pink and red are areas, as well as blue, are areas of phylogenetic endemism. And you can use those maps to understand what are the drivers of biodiversity in these regions and what kind of question and what would it look like in the next century under climate change. Secondly, we can use this data to train early career scientists in the Global South on bioinformatics. I have projects that I'm planning to use GBIF data to train scientists in the Global South on bioinformatics or big data analysis. And finally, next, we can also use this data to guide further sampling in areas where there is poor sampling or there's a limited sampling work that has been done in those regions. Next. And we need to, just to remind you that the Global South contributes a lot to our understanding of ecology and evolution. All these ecological theories listed on these slides, they were made available, they were developed because some scientists in the past worked in the Global South, understanding the drivers of biodiversity in the Global South for them to be able to formulate and postulate these theories, which we are learning or teaching in schools today. So this is just to highlight and remind us about the importance of the Global South biodiversity. And finally, next slide, just to highlight my contacts and information about current projects I have going on about the spatial phylogenetics of the African flora, and building on that to work on functional trait diverse data as well, and amassing these data sets so as to be able to answer questions that has to do with understanding the processes and patterns that drive biodiversity in Africa and in the Global South more broadly. Thank you.
Thank you very much, Israel. Next, we have Robert Kuby from the Royal Botanic Garden, Edinburgh, to talk about the use of Wikidata for recording identifiers for collecting expedition for open linked data.
Hi, thank you for asking me. So my talk is about expeditions and recording expeditions in Wikidata. And it sits quite nicely alongside Jason's talk about the individual collections that he was looking at. As we all know, objects and data are distributed across collections in many different types of institutions and across many different types of information carriers. By that I mean field notes, travel journals, maps, photographs, etc. By linking the data from these expeditions to their related objects, specimens, archive material, etc., we create a network of information that includes both data from museums, libraries, archives, and as such, it enriches the existing data in each of these separate silos. As most of you are aware, research expeditions are an important source of natural history specimens. However, as there is no global registry or identifier of research expeditions, there is no way to link this information. And this hinders the effects to enrich the historical contexts surrounding these expeditions and their collections. So that's the motivation behind our work on Wikidata and the biodiversity information standards. So next slide, please. So a small group of us data managers in a number of institutions set up an informal group of how we might start to make some of these linkages. As expeditions were an important source of specimens in all of our collections, we worked out we need to link research expeditions to the other entities within our institutions and between institutions. We knew we needed a stable identifier for expeditions, and we needed a place to store some of the metadata and the descriptions about the expeditions. And most importantly, we realized that these expeditions needed to be viewed in a historical context, as many of them were occurred in a world that was in a very different place and had very different and significantly different cultural and moral frameworks. So we had an idea of what we needed, but it was where to actually do the work. And the decision was made, much like Jason talked about, the place to do this was in Wikidata. I'm going to drop a link into the chat now. He says, hopefully, if I can find chat, there we go. So we set up sorry, new slide, please. So we set up a wiki project, and that allowed us to start to work as a collaborative team on trying to work out which tags within Wikidata we wanted to use to record some of the information that we wanted to record in Wikidata, where we wanted to record the name of the expedition, which tag we wanted to have as the destination, the funder, etc. And we created a Wikidata project to try and bring all of this information together. What you're looking at on the screen now is the schema, which Wikidata codes we use to align to each of the schema, each of the expedition data points. By that I mean, how do we record the start point of the expedition, and which Wikidata property do you use for that? So if you are interested, please have a look at the Wikidata projects, and you can start to see how we're starting to record some of this data. It also allowed us as a group to get together and apply to TADWIG, the Biodiversity Data Standards Organization, to become a task group within their framework, which enabled us to become a little bit more semi-official and start to apply to some of the EU projects and the EU work for some funding to allow us to get together in person to try and work on some of this data. Next slide, please. This enabled us, well, this is a terrible slide, you can't really see what's happening, but on the left-hand side, there is the RBGE's collections management system, and there is a specimen and a link to an expedition. And on the right-hand side, there is the Wikidata record for that expedition, saying who was on the expedition, where it started, where it finished, etc. And as our material was collected in multiple duplicates on this expedition, it allows other people to link to the same expedition. Next slide, please. Because there is data in Wikidata, it's a horrible sentence, it enables you to start to build tools to pull together some of the information. So this is a visualization using a little work in progress.
We're at 5 minutes.
Which pulls together specimens from a particular expedition, the Harmon Alaskan expedition, and it pulls data from multiple institutions. That's me. Thank you very much.
Thank you, Robert. Next up, we have Amanda Mazza, who is from the Duke University Lemur Center, and she will be providing an update from the Zumu network.
Hi, everyone. Thanks so much for having me. I'm Amanda Mazza. I am the data manager at the Duke Lemur Center, and I'm also the co-lead of the Cyber Infrastructure Working Group for Zumu. Next slide. I'll just give a little background of the Duke Lemur Center. So we've been around for about 60 years, and we have over 4,500 individual lemurs and all the data that is associated with those animals. So we kind of have the largest collection of lemurs outside of Madagascar. So some of the data we collect is anything between behavioral observations to life history details, all the way to the medical information of these animals. We also have a museum of natural history that houses our osteology collection, and that includes about 90 full lemur skeletons of those animals that had once lived at the Lemur Center. And on top of that, we maintain a large fossil collection and a growing archive of digital scans. So at the Lemur Center, we started building kind of like our own extended specimen network by integrating data from our living lemurs with our museum's osteology records. And as you can see in the slide, like many other places, our data is sort of collected all over the place. So now we're trying to find a way to hone all this data together. Next slide. And so meanwhile, I was collaborating with Zumu. So Zumu is an NSF-funded research network. And like many of these previous talks, Zumu is trying to bring together data from different avenues, specifically zoos and museums, with a mission to link the living animal data with natural history specimens, and also to try to make it more discoverable for researchers. So the idea is just to connect data across time, institutions, and disciplines. And ZooMU has 4 working groups, and one of them being cyber infrastructure. So next slide. So back in March, we hosted a ZooMU meeting at the Lemur Center specifically to talk about cyber infrastructure. And one of the big outcomes was sort of a decision to build an integrated cyber infrastructure network to support our main group's goals moving forward. So since then, the cyber infrastructure working group has been developing training materials that to help zoos and museums kind of get onboarded to platforms like GR-Cycle and GBIF. We've also been helping institutions create and publish species checklists using data from their zoological collection management systems. And so since the Duke Lemur Center has sort of taken these steps already, we were enrolled in GR-Cycle and GBIF, and we've also published 4 data sets in GBIF so far. So we have a species checklist, our osteology collection, which is linking our life history data and including like body weights of those animals, and some of our fossil data. What we're now trying to do is collaborate with the Memphis Zoo in order to help them do the same thing. So one of the big goals of our cyber infrastructure working group is to create these steps and tools to make it a little bit easier for zoos to migrate their data and to collaborate with natural history museums that may hold their specimens and hopefully get some of this information out into a platform like GBIF. So this is sort of just the beginning of what the ZooMU Cyber Infrastructure Working Group is doing. We're very excited about this initiative. We are looking to collaborate with other institutions, museums, zoological institutions, and to get more of this information connected together and hopefully out into a research centered platform, kind of like GBIF. So if anyone is interested in joining this collaboration, feel free to reach out to me. You can also check out ZooMU at the zoomunetwork.org and look around and check us out and see if there's anything that you're interested in joining and participating in. Thank you so much.
Thank you, Amanda. Next, we have Shannon Peters from the University of Wisconsin-Madison. The title of this talk is Microstrat as an Integration Point for Biological and Geochemical Data.
Hey there, everybody. How's it going? I appreciate the opportunity, and it's been great hearing from the speakers thus far. It's a neat lineup here, so thanks for everyone being here. I'm going to talk about Macrostrat, which my collaborator David Quinn and I have been working on for some time now, that we believe is a pretty useful integration point for a variety of different data sets. And of course, for this group, the biological and geochemical might be most relevant, but it extends to include economic resources and pretty much everything else we get out of the Earth's crust. So I'm going to pause on this screen here to point out the contact information, macrostrat.org, and our mobile app, rock.org. You can find us if you're interested from those 2 points. But before I move on from this slide, I just want to pause and say what Macrostrat is. It's a geospatial relational database. It consists of 2 things: geologic maps and geologic columns. And the secret sauce, I guess you would say, for Macrostrat is we've got a nice system that integrates geologic maps composed at multiple different scales in the field by hundreds of different geologists, and we put them into a single environment where they can be leveraged effectively together for new and things that are greater than the individual map regions themselves. And so over 300 maps are integrated into the system and queryable and readily incorporatable into workflows. The other part of Macrostrat are geologic columns, and this can include things that are regionally composited, like the one you're seeing here that represents a broad area of stratigraphy, in this case, Northern Ohio, or it could be specific cores or measured sections. But those 2 things sort of come together to form the foundation of Macrostrat. And within those 2 things are all kinds of other linked attributes, things like geologic time handled in multiple different ways, discrete bins and continuous ages and things like that, stratigraphic nomenclatures and the complexities around those, lithology types and hierarchical relationships among them, and so forth. All that sort of encompassed in this framework as well. And one of the things about this is it's matured enough along that we have some really quite powerful API hooks, CC BY license, so very usable in any context. And those can be used for all kinds of interesting things. All right, next slide, please. Before diving in to the linking of data through biology, I just wanted to point out why we actually bothering to do this. And this is where my science comes in. And basically, you know, I've come to appreciate that the rock record is more than a passive recorder of Earth history. The assembly and destruction of this thing drives the evolution of the Earth system in fundamentally important ways. I mean, take a deep breath. That's because of rocks and life coming together in a cool way. So our motivation is to provide a way to quantify the rock record in time and space. The way we do it happens to carry a bunch of informatics advantages. It's just the natural join for a bunch of different things, whether it's building stones in a city or fossils in a drawer. Next slide. All right, speaking of fossils and drawers, where this is a bio-oriented crowd, and it goes without saying, you know, Jason already told us about the centrality of specimens, and those are important things. Donat told us about taxonomic names, and those are one of the hooks into the collection like this. Next slide, though. But if you zoom in on the labels, you'll see that the other hook that's almost present ubiquitously in many fossil collections are the stratigraphic elements. And so here they're encapsulated by names, Cutler Formation. And in this specimen on the right, you can even see the age field is not an age, it's a stratigraphic name, Kearney Formation, Kesey Formation. And the assumption is that has time significance, and so it does. Next slide. And then, of course, this extends to all kinds of other measurements, geochemical measurements. This is just an example table from the literature where there's a coordinate involved from this one spot of a core, but it intersects multiple different stratigraphic units that, in this case, have names. And so stratigraphy is just sort of a natural join. And just an interesting observation is that science and society as a whole, I think, has a rather extractive relationship with stratigraphy. And what we're doing is basically providing a framework for the stratigraphic record itself. Okay, next slide. All right, and this will be where we end. What we are able to do is in time and space really provide some constraints on the surface expression of rock units, the age expression of rock units, a working model for age, and go from there. And so I'm just going to go through a rather regrettable list of bullet points here next that kind of highlight the detail that comes with each one of those units, lithological and temporal. And here's what we can do. Last bullet here. Go ahead, next. Yep. So what we have, what we have in place is a pretty good starting point for a bunch of regions. We have a lot of deep sea drilling data, pretty much all of it from the EODP project, a few other regions besides North America too. We handle nomenclature and the complexities around nomenclature, kind of like taxonomic names. It's a similar situation for stratigraphic names. We can handle that sort of thing. We make a lot of explicit linkages between different data sources like geologic maps, columns, and even the scientific literature. We have an age model that allows us to make very precise predictions, hopefully accurate ones, that constrain the ages of fossils much more finely than is typically possible in, say, a museum drawer context. And then we have APIs, as I mentioned, that really surface a lot of the capabilities of the system. And unfortunately, I'm not going to talk about the work we're doing with the paleobiology database and the sedimentary geochemistry project, but these things come together so nicely in the stratigraphic record, and that's what we're doing, and you have that ability now too. Thanks.
Thank you, Shannon. Next up, we have Randy Singer from Augusta University to talk about how community science can bring together natural history collections and historical survey data.
Hey guys, thanks for the invitation and thanks for being here today. I'm going to talk about some of my research program that we do here at the Augusta University. But first, I want to highlight a really cool example of some collaborative work we did when I was at the University of Michigan, which was my appointment prior to coming here. So I'm super interested. Ever since I was fortunate enough to be one of the few people that did their PhD at iDigBio way back in the day now. It feels like it was yesterday, but it's a while ago, looking at large amounts of aggregated data. And ever since then, I've just been absolutely obsessed with digging deeper into those aggregated data to try to answer really complex ecological questions. So currently, our lab here at Augusta is looking at the deep ocean and teasing apart community structure in the deep ocean, which is totally impossible to do with modern sampling techniques because you save up all this money, go out on a ship for a month, and maybe you don't even collect your study species. But luckily, we have access to our time machine of museum collections and all these great biological collections we have. to sort of explore that data, right? And so that's sort of what I'm focused on. And if you're interested in collaborating at anything like that, like tackling complex ecological problems, I'm happy to collaborate. So here I'm going to highlight one project that we worked on that does this, but actually takes a cool spin on it. So one of the biggest problems we have with our data is actually processing all of that data, right? As all of you know, you can go through a database and you could spend your entire lifetime trying to tease apart one thing, but the sheer volume of work is just incomprehensible for even you or a team of your own friends at your institution. And so what we did with this project, this is called the Changes Project, where we used the Zooniverse platform, which if you aren't familiar with that, that is a crowdsourcing platform where we took field notes, so actual field data from the Institute for Fisheries Research which is located near the University of Michigan. They have hundreds and hundreds and hundreds of these cards that have been amassed over decades that highlight ecological factors about lakes in Michigan. And here you can see an actual map of all the lakes that are in this database. And some of these are tied to specimens that are in museum collections, and some are not yet accessioned to museum collections. So this is kind of a hybrid between the two. But what we had were all of this wonderful ecological data that usually goes missing when you have the data in the aggregator, right? The aggregator is just showing presence, absence, maybe some information about species, things like that. But to get to the deeper data, you have to actually contact the collection and talk to a collection manager or something to get the data. So we developed a way to, if we got the digitization done of all of these cards, and then we uploaded them to Zooniverse, where we had over 3,200 volunteers participate in this activity to take all of this wonderful ecological data, digitize it into a standardized fashion. We processed over 140,000 objects, giving us 450,000 data points. And you can see our workflow there, because I know in this community, you guys are just obsessed with workflows. So basically the workflow for this was cards go into Zooniverse, they get transcribed by the transcribers, the data come back to us at the Changes Project, and then we, as a lovely gift wrap, give that back to each of the institutions that were involved in this process. So this sort of formed an awesome blueprint for how you can do these things in the future. If you want to read our paper, it was published in Scientific Data, where we actually now then return and use that data to ask some complex ecological questions about how fishes are moving around in different lakes, at what age are they becoming mature, things like that, how this has changed with climate change through time, and things like that. So this is just a simple example of a complex question that we used a creative set of interesting community science efforts to digitize and get them in the system. So again, if you're willing to collaborate, hit me up. I love aggregators. I'm a bioinformaticist at heart, and I'd love to work with you guys. Thank you.
Thanks, Randy. Next up, we have Jennifer Slotnick from Databra. I don't know if I said that right. The title of this talk is FAIRY Upstream Cross Repo Submission Readiness Checks.
Yeah, hi. So I'm Jennifer Slotnick. I'm coming here from San Francisco. And I am working on an open source tool. So it's a local first validator that helps researchers who are submitting data sets to repositories and cuts down on bound submissions and the back and forth between the other parties that they're working with. So I wanted to come and talk at this group because I feel like it fits into the BIOFAIR network, and we could all do some collaboration there. So next slide, please. So the tool is called FAIRY. So this screenshot on the left side is showing just an example in the command line, but the tool also has a visualization and like Streamlit if you're more of a drag and drop person. So basically, again, it runs offline. So it's a tool that you download to your machine, and you'd be working with your own data and comparing the metadata schemes to the information that needs to be reconciled. So the tool gives you a pass, fail, warn that you can set up, and it points out what has been warned or failed, and why and how to fix it. And then you can rerun the tool again after changes have been made and see what's been resolved since the last time. And this is also providing a shareable resource. So it makes a JSON and a Markdown report that you can also share between other parties to, again, help like reconcile the data that you are working with. So next slide, please. So this matters for the integrated data network. So metadata is super messy, and it's definitely something that we can use tools to reconcile. So the weird encodings, inconsistent IDs, strange dates that all need to be formatted into a consistent way can be pointed out and fixed quicker if we have a way that we can see exactly where the issues are and how to fix them. So again, it's reducing that back and forth between curators and handwritten emails and things like that, where you can just actually take this Markdown report and share it. And again, there's no portable like pre-flight way that people have been working with metadata that I've seen so far. So this tool is kind of unique. And again, it's going to be open source. So the goal is that I started, I have a genomics background. So I started with Geo, but there are many other repositories like we've heard today that you could be working with. And it's really an open source tool where eventually people will be able to write their own rule packs and exactly what data that they're trying to reconcile against. So hopefully there's going to be a community resource where you can go and write your own rule packs and communicate with other people. Next slide, please. So this is my contact information. So if you scan this QR code, it takes you to the website. We're still working on the first release, but it's coming out really soon. It's almost done. And I'm looking for collaborators in the BIOFAIR network because, again, I came from the genomics side, but I'm really interested in other domains, and I feel like we could have a nice collaboration across domains. So, if you're a microbiologist, paleontologist, any of those, I'd be happy if you reached out, and all my connection information is here as well. So please take a look. Thanks.
Thank you, Jennifer. Last up, we have Jack Williams from the University of Wisconsin-Madison. The title of this talk is Integrating Ancient Environmental DNA with the Neotoma Paleoecology Database.
Hi, everybody. Great to be here, and I've loved seeing all these talks. So I'm also representing a coalition of people working together, as Rachel talked about, very much working in this data stewardship space, gathering scientists together to share data and make it available to broader communities. And so the data integration effort that we're actively working on right now to talk about today is ancient environmental ancient environmental DNA, which is kind of one of the hot new areas in the paleoecological sciences. So this is a project with myself, Simon Goring, Jonathan Nelson at UW-Madison. Hey, Shannon, good to see you. Tricia Spanbauer at Kentucky, and then Amy Amerbo. So next slide. So what's exciting about DNA and why whereas some groups are really focusing on legacy data, and we do a lot of that too, this is a case where there's a community that's very actively growing, and the number of new data sets are doubling every couple of years. And so this is a place we want to try to get ahead of the curve and work with these groups to establish best practices in data sharing and data availability. The things that ancient DNA do is that there are whole sets of clades that are not very visible in the traditional fossil record, either through micropaleontology or macropaleontology. But DNA is ubiquitous. And so now through recovering DNA from different sediment horizons, as far back as 2 million years ago now, DNA is being recovered from Greenland sediments, and teams are working to push that horizon further and further back. I don't think we'll ever get to the dinosaurs, but we're certainly pushing back over millions of years now. And the map on the lower right shows sort of the site network, and the level of data compilation in the field is roughly that people have gone through papers, pulled out lat long coordinates, and can make dots on a map showing where these records have been collected, but there's no deeper integration and really no synthesis possible to look at biodiversity dynamics across sites, across research teams, across space and time. Next slide. So I'm one of the leaders of Neotoma, Neotoma Paleoecology Database. We work in partnership with the Paleobiology Database and other paleo resources. Neotoma specializes in the timescales that last several thousand years to roughly last million years or so. So quaternary, intermediate timescales, and the data model that we have built is really suited for storing information about biological occurrences and abundances in the stratigraphic column down depth with some time estimate attached to it. And this model that has been built is widely usable and supports different data types. So vertebrates, pollen, diatoms, ostracods, testate amoebae, lots of different groups have started using this. In our community, people organized by taxonomic groups. So we have the diatom people and the vertebrate people, each have specialized in their own domains. And so they all now are using Neotoma as a common backend to store their data and upload data to it. And then one concept that we found really useful in terms of organizing these communities, giving them, empowering them to make decisions about the data and positions of leadership, is this concept of constituent databases, where there are different groups within Neotoma, such as the European pollen database, the International Ostracod database, each one of which has its own data stewards and leaders that are uploading and curating data, but they're all using Neotoma for the common backend and source here. And we really rely heavily on human curation, this crowdsourcing of this incredible work that people have talked about, and through this network of data stewards around the world, we're able to get these data sets into Neotoma. Let's go to the next slide here. So where we are now, and this is again very much at that geo-bio crossover space, and I have deeper roots on the geo side of things. Working with environmental DNA, we're really working in the bio space, and so we have formed a joint working group of ancient DNA experts and Neotoma developers to talk about the current Neotoma data model and controlled vocabularies and all the rest of it. Talk about what needs to be extended or adopted for supporting ancient environmental DNA. There are different kinds of ancient DNA approaches, and there's metabarcoding, metagenomic, and target capture, sort of three broad different methodological categories. We're working first with metabarcoding data, which is the most prevalent. It's the cheapest and fastest of the three, and so most records have been generated using metabarcoding approaches. There are pros and cons to each of these, and so increasingly metagenomics becoming more popular especially for the deeper time work. There's a new standard that's been developed, the FAIR-E standard for environmental DNA by Takahashi et al. So what we have no desire to reinvent the wheel or create a duplicate standard. So we're working to adopt their standard and integrate it into our system. So we're doing the crosswalks and matching our fields to theirs and figuring out how to map their recommendations into the current Neotoma structure. That's where we are right now. We're hoping in the spring to begin actually uploading some ancient environmental DNA metabarcoding into Neotoma. And again, we're at the development stage. We'd love to also build out connections. We'd welcome suggestions and input. So thanks very much.
Thank you, Jack, and thank you to all of our speakers. That was a really interesting and diverse group of talks. I'm going to hand it over now to two of the other members of our BioFair team, Libby Elwood and Mike Webster, to facilitate our discussion.
Thank you. First, a big thank you to the speakers for providing a fantastic diversity of data types and projects and work that you're doing. I think thinking back to Matthew's, one of Matthew's original slides with the wheel with the different segments on there, I feel like as a community, we're making good progress on a bunch of those already. So that's really encouraging to see. And thank you to those of you that also provided some opportunities for collaboration and input and feedback and some links to resources and all of that. So I feel like we are, we're in a good place, and it's great to see all of you here. And thank you to those of you too in the audience for being a part of this. So as we're moving along here, there's a bunch of time for questions. So if you'd like to put your question in the chat, we will be monitoring that. And we could, if you prefer to raise your hand, we can also open up your mic and you could ask it live. It doesn't have to be a question, or rather it could be a question to a speaker directly. It could be to the community at large here. Whatever is on your mind. And as we give folks time to think of their questions, we will get things started. I'll put a question in the chat here. And this is for anybody, either a speaker who would like to maybe elaborate on or provide some more examples about this, or anybody in the audience as well, maybe even especially somebody in the audience that we haven't heard from. Can you share a success story or near success of integrating disparate data types in your work and what lessons were learned? So any takers on that? Doesn't have to be a groundbreaking, earth-shattering type of success either. I think the baby steps along the way are certainly worth sharing and discussing as well.
Yeah, I'll say I feel that several of the talks presented some success stories. I thought we've heard some awesome success stories, but it'd be nice maybe to highlight some of those lessons that were learned along the way. The talks were nicely polished and showed the success themselves, but what were the lessons learned and some of the take-home messages for the broader group? Robert, I see your hand up.
I was going to talk about a different project, but successes and failures along the way. Successes are, it's lovely to get together with other data managers from other institutions. So in our little group, we've got people from GBIF, we've got people from the National History Museum in Berlin. We have a Wikimedia, what do they call her? She calls herself a I can't even remember the term she uses, but somebody who's a volunteer who specializes in working in Wikipedia and Wikidata. And they just bring everybody brings different things to the table, different experiences, different ways of looking at the data. I'm at a botanic garden, so I end up pushing the plant element. We're at a botanic garden that has expeditions and has always had expeditions going out around the world. So we have brand new expeditions heading out next week. And so how do we map George Forrest's expeditions in 1830, but also map a group going out next week to collect plants in the wild? So that's been a big plus for me, just that level of interaction.
Great, thanks, Robert. Sounds to me like an important part of the process is that collaborative experience across institutions, across expertise. Jason?
Yeah.
Yeah, Mike, thanks for that question, because I wouldn't say this exploration of Wikipedia, our exploration of Wikidata is yet a success, but we're seeing success on the horizon. But some of the challenges I initially encountered was kind of learning a new community. The Wikimedia and Wikidata community are not totally foreign to me, but some of the details of how they operate are unfamiliar. Like I learned that it would not be practical to put all 9,000 Sherwin-Carlquist specimen records into Wikidata because it doesn't really rise to the level of what the community considers notable enough. So that's one of the reasons we branched off and created our own separate instance of Wikidata to be able to freely add and change those types of things. So I've been sitting in the comfortable world of Symbiota and Darwin Core, and getting into those other realms has been a fun learning experience.
Awesome, thank you. Shannon.
My comment might be a little meta or high level, I guess, but one of the lessons I've learned is for many of us who are building data structures of one type or another to reflect something or other in the world, one of the lessons I've learned in doing that is, you know, don't try to design what you wish they would be. Design the structure to what they actually are. And that applies both sort of how they're actually expressed in nature or how they're actually expressed in human products that you might be synthesizing. And once you accept that it's not going to be perfect, but you're sort of tweaking your structure to that, it makes the work of populating it, which is the whole point, much easier and more sometimes much more informative.
Cool, thank you. Donat.
Well, maybe a success story is what just recently happened with GBIF is that since 2008, we tried to create taxonomic publications as data sets and talk to various groups. But more recently, we have a fantastic success that there are about 40 journals now that produce TextPub. That's an XML schema, which includes all the metadata. It includes links to gene sequences, the digital specimens, and so on, that are published, and within a day they show up in GBIF. So it's a fantastic thing that you publish, and the next day or within a couple of hours, you can see the results in GBIF and make it accessible to everybody. It goes in checklist bank, so you know that there's a new species. It goes in GBIF. It goes in the biodiversity PMC. So it's this cross between PubMed Central and the biodiversity world. So it was a long journey from 2008 to now, but now we have it. You can actually look at this in as hosted portals, another tool provided by GBIF to highlight and present your data sets the way you like to have it. And the frustration, though, is that it looks like that the taxonomists don't really understand that the really value is that what you say, you have to link an identification to a treatment, to a taxonomic concept to make use of all this ecosystem. And so my really challenge is here, how can you convince the community in the museums who are doing all this fantastic digitization, the community that does all the barcoding, to actually go beyond just an identification, add a name, but actually add and cite a taxonomic concept to actually add the specimen to the huge biodiversity knowledge graph.
Excellent, thank you. I'll just add, since I don't see another hand up right now, I'll add that I thought from listening to the presentations, all were very exciting, but one concept that really jumped out to me and is exciting to me is Robert's concept of the the expedition identifiers. Because for me, an important aspect of the extended specimen approach is that there's a blurring of the distinction between specimens and data. And when you go out on an expedition, you'll collect some specimens and you'll collect data from those specimens, but sometimes you might collect photos or blood samples or what have you from individual organisms that were not collected. And to me, the expedition identifier unites all of those data and samples together.
That's a good point, Mike, and even kind of loops in that related notion of a digital extended specimen where even if the core isn't a physical specimen, just a record of that organism having existed and then being able to extend out from that. So that's a really good point. I see a couple things in the chat too. Hello, Elizabeth. Question to the community. She says, for those who have integrated different types of data, have you had a chance to monitor or follow up on how those integrated data are being used by broader user communities or the target community of users for your projects and lessons learned there? Anybody care to take that on? It's not an easy task for sure to have those metrics, or maybe even worth discussing then is if you were interested in doing such a thing, how would you go about it? Israel, do you want to take that one on?
Yeah, just a quick, I mean, I believe we know this already, but in terms of data usage, it's often strongly recommended that as herbarium or museum collections should at least give DOIs to their data, and even when you upload them into GBIF. And it's also something that we as end users of this data, at least to be given this, to have this awareness that when we download this GBIF data for or eBird or whatnot for analysis or statistical analysis to answer questions, to cite these DOIs so that it is a way for the owners to track who is using their data, and will help them as well for their own institutional reports.
Thank you for that. Yeah.
Excellent. Other thoughts?
Israel, I see your comment there too about funding digitization projects. I don't know if you wanted to discuss more about that, but yes, we definitely hope that there are a good number of them going forward. Fingers crossed for that.
Yeah, I know, especially because the one that at least I'm part of is digitizing herbarium records, over 2 million of them across 31 herbaria in the US, and collaborating with about 4 other African countries, I think Malawi, Ghana, Rwanda, and Gabon. But there's a lot more to be done. And especially the fact that Kew Gardens, Brussels, and Paris have a huge volume of specimens from Africa, which were collected in the 1800s and early 1900s, which have not been georeferenced, but hopefully, as we're doing this in the US, hopefully that same energy travels across the ocean so that those data can be made available as well. Yeah.
And Robert, I see your hand up.
Yeah, it was to answer the question around whether or not there's any chance to monitor or follow up on the integrated data. At the moment, what's happening is people are coming to us and asking for a particular data set or to talk about a particular set of collecting notes or a particular set of images or a particular set of specimens collected by a collector. And we say to them, you know, okay, we've got these specimens, but also, did you know we have the diaries, we have the images, we have the so-and-so. And so they trot down to a different part of the institution, look in the archives, look in the image store, and then make the connections. What the next step is to have those available so that we don't need to happen upon somebody and find them and then go, oh, by the way, did you know we have? And to have it available there within one of the different, in each of the different collection management systems, to say, click here to see the diaries, click here to see the images, click here to see things. So we're recognizing a need, but we haven't actually gone as far as to start to think about how we qualify or check that need is being fulfilled.
That's an excellent observation, and it's a great segue to another question, I think, about what sort of tools or platforms or concepts do we wish were out there that would enable the sort of seamless integration across different types of data and or different types of collections and data types that would allow for those sorts of things to happen? Any thoughts on, you know, what's missing that we wish we had? Go ahead.
Just a suggestion, being back from the Living Data conference in Bogota last week and the GLIF governing board. I mean, one of the really interesting aspects is the new Darwin Core archive, the new Darwin Core that's coming out. It's a new extended model, which includes much more. It includes like 70 terms that, for example, include sampling and others. So what I suggest to this community, if you are not aware of that, go check this out and get involved. Mm-hmm. This is like work in progress. A lot of has been done so far, but there is phase now to comment over the next couple of months. So please go there and see that you can get the expeditions included, or figure out whether the expeditions are already modeled properly. And if you have any other things, suggest this. It's very cooperative process, and if it's adopted, it has a huge effect, because there are billions of records now getting into GBIF and disseminated, leading to thousands of new publications.
Excellent. Thanks for that that comment. And I see you have it in the chat as well, something very similar about encouraging people to go and take a look at that and contribute. Other thoughts?
I see sorry, the username isn't entirely easy to pronounce here, but a question that says, are there studies that are combining traditional taxonomy of aquatic macroinvertebrates with eDNA and deep learning / AI with advanced imaging approaches?
I'm curious about that as well. I don't know.
Yeah. Or maybe even related taxonomic groups, if not.
Öppna da, bis etwa 1:30.
Sorry.
Yeah, I put that question there, Libby. It's Shea
Okay.
Tuberty. I'm sorry, I forgot to change my name for my email address. So yeah, I'm a traditional taxonomist, working with faculty in North Carolina that are also looking into eDNA. So they've advanced the aquatic eDNA science around macroinvertebrates as a means of bioassessment of water quality, and diversity analysis over a long time in climate change. And then we've got a third faculty member at a sister college that's also using advanced digitization. So she's got an inline aquatic macroinvertebrate entry system that uses advanced lasers. And she's able to look at both the taxonomic structures, external structures, but also, interestingly, the lasers reflect off of the integument of the outside of these invertebrates and actually give you information about what the skin is made from. And so she's been able to identify not just macroinvertebrates like arthropods, insects, crustaceans, but also flatworms, for example, which are really notorious for identification. You got to get them express their genitalia, which they don't like to do. So anyway, so that was an unexpected outcome, and we're really excited about this triangulation to allow folks that aren't interested into the legwork to identify things using traditional taxonomy. Maybe we can move things quicker with eDNA. Maybe we can move things quicker with visualization. And so just wanted to see if there's anyone out there working in a similar triangulation event, maybe with plants or something else similar.
Excellent question, Shea. Any responses to that question with examples of that sort of integration?
I mean, I can speak from personal experience, but only for fishes, but because it's an aquatic system, it's similar. So the only experience I have with particularly with eDNA was at least in fishes, I feel like there's some gaps that need to be addressed before it's like widely accepted as something that like can be used as a proxy for specimen collection. I actually just published a rebuttal to a big article where they were talking about how this fish was near this marina and it had never been seen before. And then by looking at the marine logs, I showed that people were just cutting fish on the dock, and it was putting that fish at the dock, right? So until we can address like actual markers that tie things to organisms that are where they're from, I mean, I think at least from the eDNA spot, it's hard. And then if we're talking about like, I think you said something about like AI or something like that, like, being able to do image recognition, that could be something. I still think, at least currently, there's no replacement for a person validating things, so I would treat AI or not the true AI in the sense of using machine learning and things like that, because the like ChatGPT and all that is not really AI. That's just a fancy search engine. But like real machine learning, I think I would treat it the way we did previously with molecular studies, right? Like you look at the expression of genes in different populations, and then you go back and look at the morphology or look at behavior or look at things to like validate. It's like a good, what's the word I'm looking for? Like pilot. Like it's a good pilot to look into something, and then it always, I think you need to validate.
That's an awesome observation, Randy. Thanks for that. I see we're starting to run short on time, so maybe we should turn it back over for some closing remarks or thoughts.
Okay. There. A little review. Oh, there we are. So it's my job to sort of sum up, and well, not to sum up so much as to just make concluding remarks. First of all, I want to thank everyone, all of our speakers and all the participants, for a really interesting selection of talks that, diverse as the topics are, I feel fit together really nicely. We did not know how this was going to go exactly, but I'm really pleased with the outcome. We could have spent 5 months planning, and I don't know if we would have gotten nearly as interesting a selection of complementary talks as we did. So again, thank you. I also want to thank the BIOFAIR team who worked on this, especially our emerging professionals who have just been amazing and so delightful to work for, collaborative, enthusiastic, creative, energetic, and if they're the rock stars of the project, then Jyotsna Pandey, who keeps us organized, is definitely the lead guitar. So at this stage, this is really our last BIOFAIR event, and we're closing out 2 years of planning and engaging, and in many cases, reaching out beyond our comfort zones. And our main takeaway is that all data communities face the same range of challenges, and that there is strong interest, and already, as evidenced by the talks today, a lot of activity in figuring out how to combine data sets and to make them work together to answer some really interesting scientific questions. I still think overall the biggest challenge is just finding the people to talk to.
Ja, een paar minuutjes. Ik heb hem half is.
And but with our funding at an end, we're not really, as much as we, after hearing this, we'd probably like to be doing more. We're not in a position to lead a future effort. Our National Science Foundation grant, for which we are extremely grateful, by the way, does not permit us to kind of lead this project anymore. But we are very interested in staying engaged and interested in efforts to collaborate with others as they arise, and to see what we might be able to come up with to continue this work. If we were asked by a group called Sortie, who were not able to attend today, they asked us to tell you that they are looking for more participation in a year-long hackathon that they're leading. It's an asynchronous hackathon about mechanistic models in ecology and evolution. So if you are interested, feel free to reach out to them. Finally, all of our BIOFAIR products, including a summary of this meeting and the actual video of the presentation, will be made available through our website. The website is here, and also our email address if you'd like to contact us for further discussions about biodiversity integration or any other topics that are pertinent to biodiversity collections. Thank you very much.
Thank you.